Okay, so just to reiterate here, our first objective is to understand what it means to be a biomolecule and how the four classes of biomolecules differ from each other structurally and functionally. A lot of what we'll see in this unit is that the way a thing looks, the way it is structured, has a lot to do with its functional properties. This is true for the body on a whole. All right, the various organs, organ systems, and structures are shaped differently, have different components, different parts, different modifications. And those structures are really important to whatever function that part of the body is responsible for. We also see this on a cellular level. So when we look at the molecules that are making up a certain structure, when we look at the components, the elements rather, that make up a biomolecule, that has to do a lot with whatever functional properties are ascribed to that different molecule. Okay, so biomolecules are macromolecules. These are molecules synthesized by living things. They're primarily going to contain carbon. And the reason is that carbon is a very stable element, right? It's a very stable atom. It's going to have 4 electrons in its outer shell, which means it can form 4 covalent bonds, which is very structurally stable. And so most of these will have lots of carbon. We're going to see other atoms, oxygen, hydrogen, and nitrogen, but in differing quantities, especially oxygen. We'll talk about fats. Fats don't have a lot of oxygen, and this is why functionally they're going to behave very differently from some of the other molecules that we'll speak about. So the 4 basic types of biomolecules would be carbohydrates, lipids or fats, proteins, and then nucleic acids. And what I really want to stress here is that although these 4 biomolecules are basically rearranging the same 4 atoms, the same 4 elements here, they're very structurally and functionally different. So as simple as the proportions and the quantities and the ways in which these elements are forming these molecules, the way that they are arranged and organized, has a lot to do with the functional properties that we will ascribe to each of these. And so fats, again, is one of the ones that jumps out. One of the unique properties of fats is that it doesn't play very nicely with water. I'm sure we've all experienced this. If you put some oil in water, it's going to instantly separate, although it's made of the same elements as the other 3 biomolecules, functionally very different. Let's start with carbohydrates. So carbohydrates are probably our main energy source. I'm sure we can all attest to this. They're probably the bulk in our diet as far as energy intake. They're composed of carbon, hydrogen, and oxygen. And there are different types of carbohydrates depending on how, again, they're organized or arranged. So we have monosaccharides, which are simply the individual monomers or units of carbohydrates. And then we can further organize those together to make slightly more complex carbs, which are disaccharides. This is if we add 2 monomers together. And then we have even more complex carbohydrates, which are polysaccharides. So this is where we get long chains, complex chains of carbohydrates, which will be much more complex, but also much more useful for the body as far as the storage form of carbohydrates. So the monomers are glucose, galactose, and fructose. These are the individual monosaccharides, and we can combine these in different ways to form some of the disaccharides. Example, if we combine glucose and fructose, we make sucrose. Anyone know where we can find sucrose, what sucrose is? If you do, you can drop that in the chat. And then we also have galactose and glucose. If we combine those things, we can form lactose, right? Which is another common household item. Does anyone know where we can find lactose? Again, if you do, feel free to drop that in the chat. All right, I'm going to keep going here. As far as polysaccharides, these are going to be several glucose monomers and even glucose, or even disaccharides for that matter, that are joined together almost like pearls on a string to create these really complex chains. One of the most common ones is glycogen. So glycogen is our main storage for glucose. This is how we can put glucose away as a reserve for energy to be used later on. Typically, we store glycogen in the liver, in our muscles, and then that can be readily tapped in whenever we need to quickly get some extra energy. So for sucrose, this is table sugar. This is the sugar that you use to sweeten your foods at home. And again, that's going to be glucose and fructose. Fructose is going to be fruit sugar. So the naturally occurring sugars that we see in fruits, those would be fructose. And then if we combine that with glucose, we would get our everyday table sugar. Lactose, on the other hand, is milk sugar. This is the sugar that's naturally occurring in milk. And then again, that's going to be basically the lactose and glucose combined. Now, how do we make these disaccharides and even polysaccharides? We're going to use a process called condensation. And condensation is basically adding the two monosaccharides together. Water is removed, and the act of removing the water covalently binds these two monosaccharides together to form a chain. So the example here is of glucose. We bring glucose, fructose together. We take water out, right? Two H's and one O, H2O. And then we covalently bind those two monomers to make sucrose. The opposite of that is if you want to cleave or split this bond and liberate these two monosaccharides, which is the case when we want to break down our more complex glycogen, right? If we're going for an extended period without food and we want to start tapping into those storage forms of glycogen, we're going to have to cleave them by inserting water. This is called a hydrolysis reaction. We're going to insert that water, and then we're going to cleave that covalent bond and liberate these two monomers and be able to use them to liberate ATP to make energy. So the condensation is the insertion of water and the joining of these two, or sorry, the removal of water and the joining of these two monomers. Hydrolysis is the insertion of water and then the splitting of the two monomers. Okay, let's move on to lipids. And lipids are very interesting. They're composed primarily of hydrogen and carbon. So what's missing? Which of our atoms are missing here? You can identify what's missing right away. Okay. So oxygen is missing, right? So hydrogen and carbon is what mainly comprises lipids. So we notice right away that oxygen is missing, and that has a lot to do with the way that lipids behave. So oxygen helps to allow for more of a partial charge around the molecule. So we can get more of an even distribution of the charge, or excuse me, a partial distribution of the charge. So it's not equally distributed. And so that's going to make molecules that have oxygen very stable, right? Very stable, very what we say as polar. Polar means that it can play nicely in aqueous environments. So if we think about carbohydrates, which we just spoke about, and then, for example, nucleic acids and even proteins, which we'll talk about coming up, those molecules can play very nicely in aqueous environments because they have lots of oxygen and they allow for partial charge, which allows them to have polarity, right? They can bond and bind in very different, unique ways. Lipids, on the other hand, are considered non-polar. They don't have a lot of oxygen, so they don't have that ability for partial distribution of the charge. Lipids are what we consider to be charge neutral. So there is an even distribution of charge around the molecule. It's not partial. And so what that means is that lipids they're non-polar. They're hydrophobic, they're afraid of water. And again, that example that I mentioned with the oil in the water is classic for lipids, all types of lipids, but that is honestly needed in the body in some instances, right? Especially when it comes to transporting substances. Our body's made of a lot of water. We talked about this last week and earlier this week. And so it's really a large aqueous environment inside the body internally. And so in order to transport certain substances, we're going to need something that is able to form a barrier with that aqueous environment. And so a lot of our transporters that we'll see are lipid made, right? Like phospholipids, for example, they help to create that barrier because they don't interact or dissolve into that water. The 5 classes of lipids are going to be triglycerides. We've also got ketones, phospholipids, which are really important for our membranes, our lipid membranes. And then we've got some more complex ones like eicosanoids and then steroids. All right, so we'll talk about those individually. Let's start with triglycerides. And triglycerides are probably our everyday fat. They're comprised of a glycerol backbone. So just to illustrate that here, here's our glycerol backbone. Okay, and then adjoined to it are 3 fatty acid tails. So each of these is considered a fatty acid tail. The glycerol backbone is a 3-carbon alcohol, so we can see that here. And then the fatty acid chains can be a lot of different flavors. They can be saturated, which is the case up here, right? And saturated just means that every carbon is bound as much as it can. There are no double bonds throughout, whereas unsaturated fatty acids do have double bonds in the middle, right? And they can if they wanted to bind additional elements, but they are not, right? So they're not fully saturated or unsaturated. And then within unsaturated, we have another subdivision of trans versus cis. So are the hydrogens on the same face of that carbon double bond? So that would be a cis fatty acid. And then if they're on opposite sides, as we can see here, that would be a trans fatty acid. What I really want to emphasize by pointing this out is that if we look at the difference in where these hydrogens are placed, that makes this fatty acid a cis versus a trans. That is literally the difference between a good fat and a bad fat, right? We know that typically our bad fats are going to be your saturated fats, and specifically your trans-saturated fats. Those are going to really be bad fats, right? And so as simple as the difference of the position of that hydrogen has a lot to do with the functional properties of the fat. Okay, so really that form follows function, that structure follows function. So the way that a thing looks has a lot to do with the way that it behaves or its functional properties. And so the bad fats like trans fats, right, for example, aldehydic acid down here are typically going to be associated with plaque formation, you know, with atherosclerosis, whereas good fats like the cis fat up here are not. And that is really the difference with the chemical makeup that will lend to that functional difference. Right. As far as ketones, ketones are typically not very important. They are usually only produced under certain unique circumstances. So really in diabetics or individuals who intentionally reduce their carbohydrate intake, which can be for dietary and lifestyle purposes. There's a new craze over ketone or ketotic diets that are lower in carbohydrates, but also as a therapeutic measure. So there are some congenital defects where removing carbohydrates is beneficial, just because certain genetic diseases, the infants don't have the proportionate or the correct enzymes to deal with carbohydrates. So it can be therapeutic or it can be beneficial to decrease carbohydrates. And these are the circumstances under which we will see the production of ketones. Now, if we hydrolyze triglycerides in our fats, basically if we break down our fats, we're going to release these fatty acids. And then in the blood, they can be converted or transported rather to the liver, and then in the liver, they can be converted into ketones. So ketones are really made as a backup source of energy where fatty acids are broken down to make these ketones, and they can be used to liberate energy. They will build up and produce a very special condition called ketoacidosis, which can become fatal because it's producing extra acid. So we know that that's going to definitely throw homeostasis off, right? Our blood pH is going to be very low as far as that acidic buildup. And then it can also lead to physical symptoms like acetone breath. So someone who is on a ketotic diet or someone who is diabetic, right? So they don't have enough insulin to actually break down their glucose, they are also going to build up a lot of ketones. And if you smell their breath, it's going to smell like metal, and it's going to taste like metal to them too. So that metal mouth or that acetone breath is one of the adverse effects or side effects of having excess ketone production, okay? Two of the common ketones are going to be acetoacetic acid. And then this is what the ketone structure would look like, right? It's going to be your special groups, and then it's going to be carbons double bound to oxygens. So the special groups can differ quite a lot depending on the different types. There's butyric acid that can make different functional properties to different types of ketones. All right. Moving on to phospholipids. So phospholipids are the third type of fat here. These have a very different property that is called an amphipathic property or amphipathic nature. They have polar heads, and the polar heads are going to be facing the aqueous environment, right? Remember here that polar means that it's going to play or interact nicely with water, whereas the non-polar tails are going to be facing the internal environment. In the case of the membrane, the lipid membrane that is surrounding all of our cells, the non-polar tails are actually facing each other. So the polar heads are facing the watery or aqueous environment, which would be the inside of the cell where we have the cytosol, but also the outside of the cell where we have the extracellular fluid. And then the non-polar fatty acid tails are going to be facing each other. All right, so this is what we mean by being amphipathic. So it has properties of both. It can interact nicely with water as far as the polar heads, and then it has the non-polar tails, which can interact nicely with fats. One of the examples of phospholipids are micelles. And micelles are structures found throughout the body. They're really important for transport. Again, we talked about the importance of having fats to be able to isolate certain structures within the body in order to transport them in that aqueous environment. And so a micelle, as we can see here, all the polar heads are sticking out. All the non-polar tails are sticking inward. Whatever needs to be transported, right? Whatever that non-polar substance is, whether it's a fat molecule, whether it's a triglyceride, it's going to be inside that micelle where we have this non-polar environment. And then it's going to be transported in this watery environment to wherever it needs to go. So micelles are a great structural molecule to help with the transport of fats and other non-polar substances. And then the phospholipid bilayer is another very important one. This is the layer that forms the cell membrane of all of our cells. And the structure of this membrane is really important, again, for separating our cells and allowing us to have an intracellular fluid compartment and an extracellular fluid compartment. So this is another reason we're having that polar head, but a non-polar tail allows for the creation of a membrane that can separate two different watery compartments, right? So we can have the internal watery component, which is the cytosol of the cell, and then we can have the external watery component, which is the extracellular fluid compartment. We also have examples in the lungs. A surfactant is another type of phospholipid. specifically in the lungs, and surfactant is made to keep the alveoli open. We'll talk about the alveoli later on, but it's basically the small little bubbles inside of our lungs that need to be open in order for gas exchange to take place. So having surfactant in the lungs is another area where having this type of unique interaction where it doesn't like to play with water is what keeps the alveoli open, because it's going to prevent that water-to-water interaction, which will allow this structure to be open and patent, almost like a balloon, instead of collapsing in on itself, which is what would happen if those water interactions were allowed to form. So surfactant is another area this is important in order to just keep our lungs open. The other class of fats here are eicosanoids. Eicosanoids are modified fatty acids. They're really important for intracellular communication. So they're derived from really long polyunsaturated fatty acids, so really long complex fatty acid structures that can have 20 or more carbons attached. Some of the more common examples are prostaglandins and then thromboxanes. Both of these components are important for cellular communication. And when we say cellular communication, we mean cellular signaling. So the structures that are tagged on a cell, or the structures that can be bound to a receptor to bring and send different types of communications from one cell to the other. So those types of molecules are really important for cell signaling and for telling different cells what to do and how to behave. Lastly are steroids. I'm sure we've all heard of steroids in some form or the other. They're really important for our immune system. They're all derived from cholesterol. And cholesterol is a ringed structure. So a lot of the areas where we'll see steroids are in our hormones, right? So estrogen, testosterone, aldosterone are some examples, thyroid hormone. All of those hormones that we hear about are typically derived from cholesterol and are these ring structures, which we know as steroids. Okay. Lastly, here are the next set of biomolecules are proteins. Proteins are really important as far as our structural makeup. They're going to make a lot of our connective tissue. All of our enzymes are proteins. All of our hormones are also formed by proteins. All of our transporters, receptors, you name it. Those are all made from proteins. On a smaller chemical level, these are polymers of amino acids. So the monomers that are joined together to make proteins are called amino acids. So these are the building blocks that form these more complex proteins. There are many, many different types of amino acids that can be organized in different ways to make different types of proteins. Again, the form follows function. The type of amino acid that is used will have a lot to do with the type of protein that is made. So in order to join these amino acids together, we must form a peptide bond. So this is a covalent bond between the carboxyl group of one amino acid and the amino group of the adjacent amino acid. So joining these two, again, releasing water. So again, a condensation reaction happening here is how we form that peptide bond. And we make a chain of amino acids, which is called a peptide. So anywhere from 2 to 50 amino acids is a peptide. So it's a shorter peptide, a shorter protein chain, whereas a much longer chain of amino acids, more than 50 would be a protein, right? So a peptide is a shorter protein chain, and then a much more complex protein is what we typically call a protein. Now, proteins can have different levels of structure. We've got a primary level, a secondary level, a tertiary level, and even a quaternary level. So this is basically the level at which we're having interactions of that molecule. So the primary level of the protein is always going to be the peptide bond. What amino acids are joined together to make this structure? That's going to be the primary level of the protein structure. Okay, and we can see that happening here by way of the peptide bonds that are formed between these individual amino acids. Now, if we think about a slightly higher level of structure, this is the secondary level of, structure. So this is where we now start to get different types of interactions among that single peptide chain. So for example, here we have an alpha helix. And an alpha helix is where we have, again, the amino group and the carboxyl group forming these cross linkages or these cross bonds that will begin to coil this chain into what we call as an alpha helix. So this is going to add a second layer onto the structure of this protein, which is that coiling of the peptide bond to create this alpha helix. Another example of a secondary structure are the beta pleated sheets. And the beta pleated sheets is another type of interaction also between the carboxyl and the amino group here. So the amino group and the carboxyl group, but just in a different fashion, right? In a different way. And it creates an entirely different secondary structure. Here we have the pleating of this peptide chain or protein chain, which creates this really interesting pleated pattern, which we call the beta pleated sheet. All right, so the secondary interactions among an already formed protein or peptide chain is going to be giving us a different structure to the protein, which is the secondary level of structure. Now, the tertiary level of structure is where we get into even more complex arrangements and configurations. Here, for example, we have a few alpha helices, we've got a few beta pleated sheets, and then we've got several of these different secondary structures coming together to give a much more complex configuration to the protein. So this is going to be the tertiary level of structure. And then lastly is the quaternary structure. And this is really the ultimate sort of putting together all the pieces of the puzzle, where we have lots of different tertiary and secondary and primary structures happening individually and then coming together to make one giant protein structure. One of the famous examples of this is hemoglobin. So the molecule that you're seeing here is actually hemoglobin. And as we can see, hemoglobin has several different components, right? We've got 2 different sites up here, 2 different binding sites down here. And so we have 4 different tertiary structures that are coming together to make this a much more complex hemoglobin molecule. Okay. Right. I'm not going to get into nucleic acids. We're going to keep that for the next time. I'm going to pause at this point and see what questions we have.